כן. לא יודע, מה השעה? זה תחזיר למרב, תסיים. עכשיו, עמי התחיל לכתוב את הבקשה, אבל כדאי שאנחנו נדבר על הניסויים שאנחנו רוצים לעשות לפני שאנחנו שולחים בקשה. אז הוא אומר לי, תשמע, אני לא יודע מה לכתוב.
חשבתי שיש לך משהו משנה שעברה.
כן, יש לנו, אבל זה דברים קצת שונים, אז זה לא מה שאנחנו יכולים לעשות. אז הוא אומר, אוקיי, נחליף את ה-NMRI ב-C, נחליף את העכברי NMRI, את הסטרינג הזה שעבדנו איתו. הוא היה מתקדים בשנייה. נחליף אותם לבלב C. יש לנו עכשיו את ה-CACVJ והייתה המלטה של מה שעברה. אז נדבר, אני לא יודע, לא דיברתי. כל היום עשינו הרצאות, אני לא יודע מה לעשות. כן, פחות או יותר החלטתי מה אני עושה כבר עד לפני רבע שעה.
לא שאלת, קיבלנו 5 דקות לפני השעה.
לא יודע עכשיו.
אבל לא אותם עכשיו, מורידים את זה עכשיו למחשב.
לא,
את מורידה את זה למחשב.
נעלה אותם עכשיו. יאללה, אתם מתחילים? אז אנחנו צריכים לדבר, מיקי, על ה...
אחרי זה, אחרי ההרצאה.
כן, אני פה. אחרי ההרצאה אני פה. נסגור את זה. מה המספר קורס שלנו? אתה זוכר? אוקיי. אני מצטער, אני לא... רגע, editing is done on. I'm adding a resource. Link to a file. אוקיי, intro. Link to existing file. כן? choose file. אוקיי. upload the file. choose the file. לא, בטח טוב. הנה, אוקיי. זהו, upload this file. מלח מים, מלח מים, מלח מים. טוב, אוקיי. In the break I will get the list of the students. I did not assign people yet to the projects because we have a little bit too many people in the course. This means you're going to have only one time to present throughout the course. And I want to make sure that everybody that is in the list is actually enlisted and wants to take the course, and that everything is in line. So I will do that today, and later today I will assign the people to the project. Okay, so this is the second and last introductory talk. Next week we're going to have Dan Segev starting to speak about single cells, single neuron computations, and we were roughly here. Okay. hopefully we should finish the basic intro. Oh, sorry. Okay, so we were talking about the two. First thing. מה שמך? Matan told me yesterday after 2 hours of teaching, who are you? Can you tell me who are you? You never told anyone who are you. So you're absolutely right. My name is Adi. I am from the neurobiology department. My lab is down here in the Silverman Institute, part of the life sciences department, and also part of the Edmond and Lily Safra Center for Brain Sciences. Okay, so I apologize for that. And maybe I'll also write my email somewhere, or you know my email. I'll write my email, my contact information. So I'm Adi, and in my lab we work on brain plasticity as a main theme. And specifically, our lab deals with olfaction and recently also audition. So we work on two sensory systems in the context of plasticity. And I'm not sure I will touch upon the work we do in the lab discourse, but I'm sure you'll get, or maybe I'll talk about it when I talk about olfaction. Okay, anyway, so last lesson we were talking about two main sub-themes in sensory systems. One is perception, and the other is sensory physiology. And we started with perception, and we left things here, roughly here, with these, the way that we are quantifying perceptions and these psychometric curves. And I showed you a curve, which is a classical way to quantify perception. And we would, for example, define thresholds by the 50% chance to get an answer correct. And then we talked about some biases in perception and behavior, because some subjects may have a bias to shift the curve to the right or shift the curve to the left. And we gave example, if you are extremely careful about giving an answer, let's say this is well known also in the textbook. It says that these things were developed in engineers who were writing code to detect bombs or airplanes coming into the territory of the country. So you don't want to make a mistake. Just yesterday there was a mistake in Eilat, a siren went off, and this was a false alarm. So if you want to make sure that your false alarm, that the price that you're going to pay, if you're going to say something happened and it didn't, is high, this is going to cause shifts to these psychometric curves. And on the other hand, if you know that the price is high and that the reward is high and that the price that you pay is low, then you can loosen things a little bit, and if you think something happened, you will shift your decisions. The question arises is, what is the real threshold, and can we quantify it? And also I raised the problem of that quantifying perceptions may also be difficult in experimental animals, which cannot really, or there's a huge gap between how humans can report on perception and how animals can do that. So the question is, how can we quantify perceptions in animal? And the answer, which still holds today, it's since the day they started with it, is predicting discriminability using probabilistic statistics. This is true also for experiments with quantifying that human are reporting the perception, and also animals. And I'll give an example from animals because this is something that we also do in the lab. This is normally using probabilistic statistics to quantify perception or discriminability specifically is possible only when we are quantifying a behavioral task, which is a yes-no task, or a two alternative force choice task, which is the acronym here is 2AFC. In mice, for example, we can ask in olfaction, this is one olfactory task that is well known in our field. You know, you can teach a rat or a mouse in a task that is called, in this case, a go-no-go task, which is sort of a true alternative force choice, some variation of a 2AFC. Basically, a rat breaks the IR beam, and then you can give him stimulus, and if it's the right stimulus, he has to go, which means to lick the water nozzle here, and if it's the wrong stimulus, let's say it's an odor that informs him not to lick the nozzle, he will retract his head. Okay, if he will lick, he will get normally punishment. So basically we can quantify 4 different choices in these types of tasks. Okay, this is a yes. So let's say this is an experiment we actually do in the lab, in an auditory system. The mouse comes in, it blocks the IR beam, and he has one of 2 options is played to the mouse. Either a target tone or a non-target tone. Okay, target means it's a safe tone, he can lick. Non-target means do not lick. And then the mouse can do one of 4 different things, or one of 2 things, either to lick or not to lick, but in the 2 different scenarios. Okay, so if it's the right tone and he's allowed to lick, he will lick, and we can quantify this as a hit. If it's a safe tone, it's a target tone, and he doesn't lick, so we quantify this as a miss. If it's the danger tone or the non-target tone and he licks, this is the worst. We call it a false alarm. Okay, because he reports that it was the target tone, but it was not. And if he hears a non-target tone and he doesn't lick, this is called the correct rejection, CR. So in these types of go-no-go tasks or 2 alternative force choice tasks, we normally end up with 4 different results that we can quantify. And then it depends on the specific behavioral task. You know, you can reward him with water or punish him with a timeout. In our case, it's an air puff, but it doesn't matter for the sake of the argument here. Okay? So in these types of yes-no tasks or go-no-go tasks or 2 alternative force choice tasks, we can end up with 4 different results. Okay, maybe I'll go back to this just to show you that this is actually works. This is data from a real life from the lab. So in the beginning, mice do about 50% chance. We quantify here, in this case, the correct ratio versus the incorrect ratio. How many times they got things correct versus incorrect here. Okay, how many times they got the hit and the correct rejection, that's the correct. And these are the 2 incorrect ratios out of the target and non-target presentations. And they start by doing 50%, but after getting a few punishments, they learn very rapidly. And the correct rates go up. This is 7 different mice, and some even get almost 5 repetitions, and they already understand the task. But most of them, it takes them time, and after a few hundred trials, they are very good at the task. So they make correct responses and very little non-correct responses. The task specifically here is to give them an easy task, which is to differentiate the target stimulus and the non-target stimulus by 1 octave. And then with time, every few days, we decrease the distance between the target and the non-target tone. And the tasks become increasingly difficult until they reach their JND, their just noticeable difference, which is basically a report of their the maximum perceptual threshold that they can achieve. Okay, so we can quantify this in animals, but the correct way to quantify this is using probabilistic statistics, and we do this by plotting the 4 different options that I showed you earlier. Okay, so we have the stimulus stimulus can be, let's say, red or blue in this case. This is the example you get in the textbook. And the subject has to say whether it's red or blue. If he says red, it's a hit. If he was shown the stimulus was red and he says no, or he says blue, it's a miss, etc., etc. Okay, and then this was done for 200 times. 100 times they played the target stimulus. 100 times they played a non-target stimulus. Of course, totally there's also 200 responses, but these will be distributed in these types of plots of hits, misses, false positives, and correct rejections. Now, the way to quantify this is to assume probabilistic or Gaussian distribution, Gaussian probability of the different options, and plot a decision boundary. Everything on the right of the decision boundary would be red in this case, or in our case, the target stimulus that I showed you before. Everything on the left would be the non-target stimulus, blue in this case. And the answers, the hits and the misses, will be plotted for the red as a Gaussian distribution. Okay, the hits and the misses. And the correct rejections and the false positives will be plotted for the blue or the non-target stimulus as a Gaussian distribution. Okay, and both of these curves will be plotted on this decision boundary graph. Now, if you can assume that if the subject makes always the correct answers, so we'll have 0 correct rejections and 0 misses and 0 false positives, these two different curves will not overlap. So this red curve will be shifted to the right of the decision boundary. This one will be shifted to the left, and there will be very little overlap between the curves. Okay? Now, in reality, when we get closer to perceptual thresholds, we make mistakes. Okay? The harder, the closer we are to the threshold, the more mistakes we make, the more misses and the more false positives that we make. And then these curves will shift one towards the other, and they will start to overlap. And the way that we quantify the discriminability of a subject to these two different choices and these two alternative false choice paths is a measure called D prime, which is basically the distance between the peaks of the Gaussian distributions of the correct or the answers for the red versus the answers for the blue. Okay, and the overlap between them will change depending on how the performance of the subject is, how many misses and how many false positives he makes. Okay, and of course you see that an ideal observer will have the exact overlap spot on the decision boundary. And if we have an observer which is not an ideal observer, which will shift either toward making more mistakes or making less mistakes, or being more loose in terms of making the right hits, this can also, both of these curves can shift to the right. But this will not affect the discriminability of the subject. So the discriminability, or the D prime, is an accurate measure of how the subject can discriminate between two different possibilities in a two alternative false choice path. Okay? Any questions? Sure.
These things do change with training, right? I mean, like
Yes.
the mouse makes a lot of mistakes
Mm-hmm.
and gets a lot of punishment, and it's like the senses get sharper.
Yeah.
And where do you say that JMD is there where they don't change anymore?
Yeah, JMD is when you give two stimuli that cannot be perceptually distinguished, so the animal simply cannot make any distinguishment, and then the D prime will be 0 basically. Both of them will be spot on.
And can they change anymore or not? I mean, if
No, no, there is, okay, we will talk about this, I think, further on, but there is, at some point you reach your threshold that you cannot discriminate anymore, and then, you know, whatever I give you the task, if you cannot discriminate it, you will perform a chance level. Okay, D prime is normally between 0 to a positive number. It's a dimensionless number, but it can also be negative, which is a little bit counterintuitive, but sometimes you will make maybe a negative D primes means that the signal that the animal should detect, he can detect it, but for some reason it gives you a wrong answer. So either something is very wrong with your paradigm, or there is some hidden intentions of the subjects to lie to you or to deceive you intentionally. So you can get values of D prime which are negative. But I don't want to go into the mathematics of this. Not that I can plot all the equations on the wall anyhow. But this is something you guys are going to do in Reshatot Bet, I think, a little bit more seriously. There's some variation on this, which is called the ROC curves. Probably the people from psychology know that. But this is just, again, to give you on the tip of the fork the types of things that we are interested in sensory physiology. Okay, we're still in the realm of quantifying perceptions. Okay, everything's clear? Okay, so just a few notes. False positives, of course, increase when noise exceeds response thresholds. You make more mistakes when it's harder. And D prime provides the separation between the means of the signal and the noise distributions. D prime can be thought of as the sensitivity of the detector also. And the higher D prime indicates better performance, so a signal can be more readily detected. And it's an accurate measure that compensates for the differences we talked about before. Okay? It's the real discriminability measure, and it compensates for differences in task demands and response criteria, and these are called the shifts from an ideal observer. No. Great. Okay, another aspect of quantifying perceptions is time. Time is a very useful quantification of perception, and basically we can say as a general rule of thumb that there is a trade-off between speed and accuracy of a task. So when you are giving more time to perform a given task, you will perform better, and this is one very good measure both in animal studies as well as in human studies, how fast can one get to the answer. This is again a classical paper from olfaction, which I may or may not teach. I still didn't decide what to teach on the olfaction, but basically you see here how long it took the animal to sample in this task. It's the same task that I showed you before, where the rat puts his nose in this IR beam and gets it's a go no-go task. And basically, if the mouse took him more time to sniff the odors, the accuracy of the responses was higher. Okay? And if you take task difficulty, which is harder, so the red curve is a harder task. So let's say to distinguish between odor, a mixture of odor A and B, where A is 60%, B is 40%, versus an odor which is 60-40, it's a very difficult task, okay, to differentiate between two different odors, while the blue task is, the blue curve shows the task of distinguishing between an A and B, which are two very different odors. And as you can see, there is a clear speed accuracy trade-off, so the more time the animal has to sample, the better he's doing on the task. There's also an issue of absolute values which decrease as the task difficulty increases, and it doesn't matter how much time you allow the animal to try to figure out what is the correct answer. Still, if the task is much more difficult, you will get lower absolute values. So this measure of time can be critical, one in assessing difficulty, and it's a very useful measure that you will see throughout the course, or in some cases in the course.
This is not a trade-off.
What?
This graph is not a trade-off.
What do you mean? If
It
you
does
get,
not represent a trade-off of speed and accuracy.
why not? If you have more, if you're doing it very fast, it will be less accurate. That's my point here.
The more write-out is more fast.
Yeah, no, this is slower.
Okay.
This is order sampling time.
It's less accurate.
Of course, it's more accurate. No. Each curve shows the speed accuracy trade-off. Okay? And if you look at different tasks, from hard, which is the red, to the blue, harder tasks, if you do it in exactly the same time you spend looking at a harder task versus an easier task, you will do better on the easier task for the given amount of time. Okay? So this is one measure of how long does it take the subject to perform the sensory or the specific task at hand. And this is a classical concept in sensory perception. The other thing that has been used very successfully is the famous example of Van Friesman. And the task here on the left is to find the blue cross. So there are three different tasks. You see on the left is quite easy. You can find the blue cross rather quickly because the blue cross has a different color in this case than all the other objects in the whole array. The task becomes a little bit more difficult. It takes more time if you now ask if all the colors are the same and you ask the subject to identify the blue cross in these types of tests. And it becomes even harder if there's a mix and match between the structure of the objects and the color. Okay, these become increasingly harder. And again, the task is the same task to identify where is the blue task. Now what Anne Prisman did, she measured how long it took subjects to identify the blue cross in these different assays. And what she found is that the time it took the subjects to identify the blue cross in these types of tasks is independent of the size of the array. So if I ask you to identify the blue cross in 1 line or 2 lines or 4 lines or 50 lines, it doesn't matter. The performance is the same. So this is a graph plotting, in this case, the number of items in the array and the performance or the time it took to the subject to identify the blue cross. And you see that it is independent of the number of items in the array, and you get a flat plot. Okay, so this is the first experiment that she did. The second experiment that she did in the harder tasks was again to identify the blue cross, but now what she found is that it took the subjects more and more time to find the blue cross in the plots. And the conclusion from these experiments is that subjects use different strategies to search for the blue task. Here we are using something that is called a parallel search. We look at the whole array as one object, and we can easily identify this thing. These are called the pop-out tasks. And the blue cross pops out very easy, and we can do the search as a parallel. It doesn't matter if the array is small or larger. We can find the pop-out very easily and very fast and reliably. Here we are using a different strategy, which is called the serial search. And we, in fact, go and search serially for the cross on each line. We are looking actively for the details of the cross. She found that it takes about normally people takes them about halfway through the plots on average that they find to search. They don't search, if it takes you 2 seconds to search the whole array, it will take you 1 second on average to find a cross. And the larger the array is, the more time it takes the subject to find the plot. So this is a different type of behavior that Ann Treisman found for these two plots. These two plots only show whether it's, to prove this issue further, whether there is a cross or whether the cross is absent in the array. In order to know if the cross is absent in the array, you can't do it halfway through, of course. You have to go throughout, you have to search the whole array, and this takes time to exactly twice the time that it takes to find the cross in the array. Yeah.
She found it by eye tracking, or how did she?
No, how long?
And serial.
No. This is what she concludes that for that the search is. Okay, we're now talking about perceptual strategy, if you will. If you do a pop-out task like that, and this is the task, it's easy. The larger you go. Now they took it further. These types of tasks. The person who took it really further is Shaul Hofstein and Arav Achisau with their very famous reverse hierarchy concept or theory, which Shaul himself will teach you about that. But the idea is the following. So in pop-out tasks, we see the object as a whole object. Okay? And this is done in higher brain centers and not in lower brain centers, not in areas like V1 or V2, but in higher areas. We start to see these figures or objects, and these are actually done first, and these are actually done faster than identifying the small details in the image. So Shaul and Narav developed this reverse hierarchy that higher things are done faster, and if you want to see the details in the image, then you go back to the lower levels of the sensory system to start to identify the different, if it's a line, if it's tilted this way or that way, and this is actually done at lower levels, and it takes more time to use information from V1 perceptually than it is to use, so to speak, things in higher brain centers. Okay, but this again, we will get back to it when we will study vision, and Shaul will talk about this more carefully. But again, the idea here is to get a picture of the general issues. Okay, so we started with the initial thoughts of the philosophers, and we started with psychophysics, and we talked a little bit about some basic concepts or I don't know, definitions, if you will, in psychophysics. Later on in the development of sensory physiology, historically, came in the topic of sensory physiology, which is the relationship between the stimulus and how it is processed by the brain. Like I told you, these can, eventually we need to combine these two so we can say that we really understand how the percept is being encoded by the brain. By far the dominant strategy in sensory neuroscience or sensory physiology is simply to follow the sensory information, the flow of the sensory information. Okay, and we first ask ourselves, what do we need to know and in what detail in the sensory system? Okay, so you tell me, what do you think are now you have a sensory system and you are a sensory physiologist and you want to start and study how a brain circuit, a neural circuit, processes sensory information. What is the information you want to achieve? Let's start. You get now a network. I give you a tissue, biological tissue, I don't know, of a frog. You can do whatever you want. What's the dream experiment? You want to understand how this thing works, right? Where do we start? What do we want to know about the circuit? How does the circuit process information? What?
Show it something and follow the flow of potential through
Okay, so you're going to start
between the presenting of stimulus toward the response of the frog.
Fantastic. So you're first gonna design some kind of stimuli, some kind of input, and the basic thing in sensory physiology is to establish an input-output function. That's what you want. The first thing. Okay, what did you put in and what is getting out of this black box? So the first thing is input-output function. Now what else? Let's say you have the input you have because you control it. Some of us have it better than others. Okay, if you are a visual scientist, neuroscientist, you have fairly good control of the stimulus. If you're an auditory neuroscientist, you have a fairly good control of the stimulus. If you're an olfactory neuroscientist, so it's a little harder. Okay? But we know what we're giving, or we think we know what we're giving if we have the right equipment. And we can measure, for example, the output. What else do we want to know from a circuit? Which may be important for us. Yeah.
Maybe a time course, how long it takes?
Yeah, this exact snapshot of the physiological behavior of the different cells. How many different cells do I have in the circuit? What types of cells do I have in the circuit? Things of that source will. These are the things you're going to get exposed to, and each sensory system has its own different players. Its own different players in the real world, in the physics of the real world, of what types of stimuli they are detecting, the type of receptors that are the players, the type of the networks that are part of the game, etc., etc. Okay? So when we're talking about sensory physiology, we need to know many things. I just plotted here, things that I wouldn't want to know. First of all is the input-output functions, the cellular physiology, like you said, exactly what is how does the response look like. If we have several neurons and cells in the network, would like to know the connectivity between the cells, the different cell types, whether these are excitatory or inhibitory cells. It's extremely important. Whether location matters, you will see that in the lower parts of the sensory system, location matters very much. This is notable in the classical tonotopic map.
Location of the stimulus or
Location of the neuron on the surface of the epithelium, things like that. Like I said, the properties of the synapses, and also this can be. It then gets more complex when you start to plug in a behavioral context and dynamics and things like that. Okay, but we will start very low and very slow. But again, I'm not sure. Every sensory system that the teachers will talk with you is going to start low, run up through the system, and get hopefully into the more complex, the more advanced stuff. I just like this. This is not something that many computational neuroscientists will put on the board. So excuse me, my being brave or stupid, I don't know. Anyway, I think we are slowly getting into the era where we are learning from people who do genetics and cell biology on how to do experiments. Because in developmental neuroscience or genetics or cell biology, where we have made really great advances, the experiments are rather straightforward. You know exactly how to approach a system, and there are classical ways to do it. First, you need to identify the building blocks, and now this has been solved. Okay, you guys are born, so to speak, into science where the book is written, and you can go on the web and get this information. In neuroscience, it's a neuron. That would be the functional unit of the system, and this is why Dunn starts this course. Okay, he starts with the basic functional unit of this biological tissue is the neuron. Perhaps people would say a small group of neurons, and we have billions of these neurons in the brain, and that's why it's a very challenging task. The next level is to study, information processing. This is where physiologists kick in. In case of genetics and cell biology, it's how these proteins interact, transcription, translation, degradation, post-translation modifications, etc. In neuroscience, it's spike trains, excitations, inhibition, plasticity, neuromodulation. This is the language that we speak. In terms of experiments, geneticists and cell biologists have been in the ball game of doing serious science for many, many years, and these are gain of function and loss of function experiments. You simply plug out a single gene and see what is the impact on the behavior of the cell, etc. And you do experiments where you do recovery, and it's very straightforward the way of thinking. Until recently, we didn't have these kinds of tools to knock out specific cell types or neurons or activity from the circuit. And this is now, we have been doing this with pharmacology and lesions for many years, but recently genetics and other means of combining optics and biology has proven very powerful to get ourselves into this realm of type of experiments. So what you will see again throughout the course is more modern neuroscience of people that are actually doing gain of function, loss of function type of experiments. And this is, again, this is not an invention of the neuroscience community, and this is the easy way to interpret what you do in an experiment, okay? To plug out a single component and in an elegant way, and to see with a fine scalpel and to see the impact of this type of experiments. And in that sense, neuroscience is lagging behind. In many senses, we're lagging behind. This is really a black box, and you'll see also in the course that we really understand only some basic functions in sensory physiology. Okay, so, wow, this is very sad, huh? 64. Okay, sensor information is gated into the central nervous system by receptor cells. Okay, thus receptor cells provide the first neural representation of the external world. This part of the nervous system is best understood by far. I was in an auditory meeting 2 weeks ago before the semester. Many, many, many talks, posters, thorough understanding of the cochlea and the hair cells and how stimuli are transformed into the neural code in the ear. I can say, you know, it's impressive how we do understand these parts of the system. This is true for the retina and early, for example, in the visual system, in the retina, and as you go deeper and deeper into the brain, it becomes foggy and foggier and foggier, and it's really the time for us, you and us, to solve these problems, and there's, of course, a lot of things to do. And again, the information from the receptor cells flows in serial parallel streams along the way where information is transformed. It's even worse up there in the CNS because many things are recursive. So you start getting into loops and feedback loops and things like that. And this is much less understood in sensory systems. One last thing before I take you, you get your break, is that, which is an important thing because many of you are not biologists, but in biology we have, you know, we look at the biological tissue as, you know, as a bunch of cells doing something together, which is what it is at the end of the day. But if you look at, you know, at the kidney, if you look at the heart, there have been really great advances in these topics, and mainly because it's much more simple, because many of these systems are a syncytium. So the same cell will do this very similar function. If you understand how a single muscle fiber twitches, the step to how a bunch of muscle fibers twitch together, you know, there is a jump, but the jump is not so big as it is in sensory physiology. Sensory physiology, like I said, neurons are extremely heterogeneous, many, many different cell types, and the task is really not easy. This is again the reason we start with Idan, which shows you how the basic functional unit of the brain functions. So let's take a break, and then we'll start talking about receptors and some physical limits of the world. And I hope yeah, it's up there.
No, you need to.
I did upload files. Syncytium. Syncytium is it's a tissue where all the players are doing the same thing, like the muscles. Okay, if you take out the muscle fiber from a syncytium, ברקמות כמו רקמת שריר, או רקמת אפיטל, כל התאים המוגדרים. קוראים לרשת הזאת של התאים, זה השם שלה. אבל הרקמה במוח היא לא זאת. המוח, כל תא יש תפקיד אחר, וכל אחד עושה משהו אחר, וכל אחד
קשה לנבא את הפעולה של הרשת.
לא רק זה, אתה צריך לדעת מי עושה מה. רגע, מה? בואו נראה ש...
אני עושה את זה בעמוד ה...
אז רגע, בואו אנחנו נגמור כבר, אני חוזר לך כבר.
נראה לי שאתה צריך את ה-choose.
מה? choose? upload the file. מלח מים, done?
לא, לא upload the file, choose.
I did it.
לא.
טוב, תגידו לי מה לעשות. זה ההרצאה.
מה הוא כבר עשה?
כן,
אתה צריך לעשות נגד קודר או something, אני לא יודע.
תגידו לו.
אתם יודעים?
הוא לא יודע, לא רוצה פה.
אולי להצ'וז שם באקשן?
אוקיי. עכשיו
If you do choose file,
כן,
here,
save, כן. Save and return to the course.
you can see it, I did it too.
That's it, it's up there. תבדקו.
כן.
או שלא. כן? יופי, צריך להקליט.
off ולא out. אחלה. כן, בטח.
סתם, רצית שציירת את שאנחנו הרבה יותר יודעים לשים על הנושא של ההסברה, של הקבלה, של הכסף, וכוחים וכדומה. אז אני חושבת שזה באמת, כאילו, ברור שהמשך הוא באמת יותר מסובך, אבל בגלל שאנשים ניחויים מסוימים יותר תפקדו, כבר למה זה דווקא יותר באמת מובן, האם זה רק בגלל שההמשך יותר מסובך, או שמשם באמת מתחילים
אני חושב שזה עניין של experimental control. איזה שליטה יש לעשיין על המערכת שהוא חוקר. ברגע שאני יכול, המערכת מתחילה ונגמרת של כף הידיים. שליטה על היצורים השונים. יכול לשנות פרמטרים. קל במובן זה שאנחנו מכירים את השפים שלנו, לא קל מבחינה טכנית.
כן, לא, ודאי.
יותר קל לרשום מניורון בבן אדם מאשר בניורון בעכבר. אז אין לי access למה שקורה שם. יותר קל לרשום מקורטקס מאשר מפוטורצפטור, זה מסובך, אבל ברגע שאני מצליח לעשות את זה, אז זה הפוטורצפטור, הוא מתחיל בפוטון שלו ונגמר בפוטנציאל הממוגן. צריך לחקור מה שיש בפנים. אנשים חוקרים מה שיש בפנים, זה מתחיל ונגמר בתא. אז לא יודע אם היית בהרצאה של באופנים. אז מוצאים ניצן של איזה זבוב שחסר לו איזה גן, ועכשיו כבר האינפוט אאוטפוט פונקשן נראה את הקורס שלו, אבל במקום לתת תזובה כזו יש תזובה כזו. נתחיל להביא תוך כ-30-40 שנה, מפענחים את כל הרשת ומכירים את כל השחקנים, במקרה הזה דווקא השבוע יהיה. השבוע יהיה, נכון? אד רובל.
כן, זהו, אבל מאיפה שמעתי אותו?
לא יודע, אלי דיבר איתי, אבל הוא מישהו שחוקר את לא את הקוקליה, אלא חוקר נוקליוס, תחנה אחר כך, או את המידיאל סופרהולמוס.
הנוקליוס למינור.
נוקליוס למינר, whatever, איזה תחנה אחת אחרי התחנה השנייה הוליסית פנימה.
אוקיי.
ושוב, אז שם יש מעט מאוד תאים, יחסית. הם מסודרים כמו חיילים בסדר מסוים שאפשר לנסות להבין מה הם עושים. וזו הסיבה, אתה יודע, זה לא בגלל שניסו פחות, אני חושב. זה בגלל הכוח, בתור הנסיינים, הכוח של הנוקליוס משכנע.
כן, זה לא, פשוט הבנתי שיש כל מיני מגמות ככה שמובילות למחקור דברים כאלה, אבל כן, אני יכולה להבין.
יש, תראי, יש סיבות חברתיות. כל הנושא של biomedical sciences, יש המון כסף למחלות, והמון כסף לדברים שקשורים ב-biomedical sciences. מאוד קשה למצוא כסף לדברים אחרים. וזה גם כבר דבר שמוביל. עכשיו אובמה החליט לעשות X או לעשות Y, זה משפיע על דברים האלה. אין חוכמה, אין חוכמה. אנחנו צריכים כסף בשביל לעשות ניסויים. אז יש דברים שהם קצת יותר גדולים ממה שאני אדבר עליהם. גם מובילים דברים ניסויים.
כן, אוקיי.
זו דוגמה דווקא יפה, ה-Computation Neuroscience Community כאן, זה משהו שצמח מלמטה, מה שנקרא, מאנשים שמאמינים שבצדק, שחסר תיאוריה ב-Neuroscience, והקימו את המרכז הזה, היו להם הרבה מתנגדים, זה לקח המון זמן. אני חושב שהיום, רק היום, אחרי איזה 20, 30 שנה, 200 התחילו, 20 שנה, 20 שנה, אז אנשים גם ברחבי העולם מבינים, באוניברסיטאות אחרות, מבינים שזה באמת משהו מאוד חשוב, דברים מרכזיים וכו'. אז אתה יודע, מאוד קשה, במיוחד עם מדענים, כל אחד חושב שהוא עושה את הדבר הכי חשוב בעולם, אז זה כבר היסטוריה. טוב, יפה. מה? מה?
לפעמים שעה או מחר. קצת נראה טוב.
אני לא יודע אם אני אספיק מה? גזר?
הבת שלי בכיבוי ארץ.
אם אתה מוציא את
זה תל אביב.
מה, יש הרבה דברים בהרצאה שהם
מה, אני לא יודעת מה אני אעשה, אני לא יודעת
אני
יפה.
לא כך אוהב.
זה.
זה...
מה, סתם, זה מרכז שאמרו לי, כאילו, לא יודעת. לא, סבבה? הרבה יותר מהכל. כאילו, זה כבר לא פוריסה, אבל...
זה קשה, כאילו אתה מניח חוקים.
לא, לא, לא. אלי עכשיו יצא מהכיתה.
התואר ראשון של הפסיכולוגיה.
אין קורס כללי.
זה גם עכשיו נראה לי סתם למדתי ציור, היה לי ציור מאוד קשה.
טוב, אני הולך לעשות קפה.
עכשיו אתה טוב, תסכים.
מה אפשר לעשות ככה?
לעשות מה שרצוי. ורצה הייתה הרבה תקופה שעשיתי תל אביב והכל, אז כאילו, שינוי כזה גדול. גם הים, אני יכולה לראות.
וכל האנשים מסביב. כאילו אמרתי לה, אפשר ככה במרכז, מרחב פתח תקווה זה קאטו מונחות המונים.
בתור פרס
הלכנו
לכל...
למרחב העילית, כן, כן, לעשות את הטיול.
טוב, אתם רוצות שם? זה סבבה?
מדהים, יש לנו כאילו מדינה מהממת.
וואו.
כן,
טוב, אני חייבת
זה יהיה עד סוף שנה, אבל...
אבל אני כאילו הקטע שאמרתי, כאילו אני הסתובבתי בתל אביב ועשיתי נסיעות במרכזי סטודנטים שעושה את זה, אבל זה נראה לי מטורף. מעבר לזה שכאילו אני גם צריכה להשכין קריאה כל...
הנסיעות והמגורים שם יותר יקרים, כאילו.
כן.
זה לא כמו לגור בירושלים ולנסוע לתל אביב לעבוד, שאז את מרוויחה יותר כסף מתל אביב, ואז גם כשפחות גרנו זה אבוד.
כן. זה יוצא מינוס.
כן.
כן, ככה זה...
אבל מעונות זה בטח לא טוב. עכשיו
לא.
אני מבינה גם למה את כל כך נהנית מהעיר.
זה לגמרי, אני חושבת שזה... זה משהו, זה לא בריא, ואז תישאר, זה לא בריא.
בדיוק.
חייבים כאילו לנסוע לזה, לנסוע,
Right, 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 right.
טיול
Close
בשפה.
notes from
כן,
somebody?
לא, אתה יודע,
Is there machine generated?
כן, ואף אחד לא עושה את זה מעבר לשנה
כאילו,
ראשונה.
זה נשמע, זה לא תקן
50
אותי, יש
hertz.
פה משהו, כאילו, יש פה משהו...
What computer? The machine noise, why is it 50
כן,
hertz?
כאילו, רוב ה...
זה נכון.
So what are we saying that it's just like
אני חושבת שזה גם באמת
the system is dead? I don't know, like electrical.
לא בא לטפל.
כן,
I think that's aliens trying
לא,
to
אבל זה לא...
communicate.
אתה הודי, אז קשה לך לחפש עכשיו ישראלי.
כן, זה גם אני לא, אני סתם...
כן, כן. זה ישפר לך את הסיטואציה.
זהו, אמרו לי שבאו
Quantifying
בירושלים זה
area.
כל
המאמרים
But I thought that movies out and show up.
כן, וגם את
בשנים
יש איזה מאמר קוגניטיבי שכתוב שזה להיום. לא שאני לא מתכוון לקרוא את זה.
Unless you're like plausible deniability. לא אמרת לי שום דבר.
בכל מקרה אני הייתי מדפיס את זה. Wait, London? לא, לא שלחו שום דבר.
It's called, yeah, it's called,
כאילו עצמותם לא ממש
לגבי בספר הנביאים, שנים. מה, אז...
Yeah, I'm gonna get a pen that's really thick ink. I can't handle these like the dry ones, but
Okay, let's go.
כן, נכון. בסדר, אז אני גם לא אצטרך... כן, גם אני בעיקרון. מה זה?
Okay. So let's try to להכניס לגיל גבוה, איך אומרים באנגלית? Switch into higher gears, not up fast enough. So we'll get some mileage. Okay, so first of all, we start with the receptor. Sensor physiology starts at the level of receptors, and the physical variables that we talked about last Wednesday are sensed by the array of receptor cells of the particular sense. The receptors are normally situated in a tissue which is called sensory epithelium. This can envision it's the retina, in audition it's the basilar membrane and organ of Corti, in touch is the skin, the receptors are located or exposed to the external world on the skin, in smell it's the olfactory epithelium, in taste the tongue, etc., etc. There are different types of sensory epithelia. You will get to know quite well the cochlea and the hair cells in audition, which is a one-dimensional array of information, which is sensitive to pressure waves in the air. The retina, we also talked, I think this was the first slide when we talked about the slide stolen from Peter Latham and Eddie Nelken of a 2D matrix which has to be transformed into a something else. So the retina is a 2D matrix, the neurons on the skin are also 2D matrix, etc., etc. Changes in physical variables cause changes in electrical potential. This is also a very basic general principle. Okay, what you can measure in the receptors is called receptor potentials. However, sensory, the stimulus itself of the energy of the stimulus itself is not the same energy that is transformed into electrical energy. They're not equivalent or they're not equal. Okay? So it's not the stimulus energy that is transformed into electrical activity. Eli used to give this example to actually calculate how much energy. So perceptually we can identify a single photon. Okay? And the energy of the photon is 10 to the minus 18 joules. And Eli would do the full calculation. I saved you the pain here. And just tell you that if you measure the result of the photon hitting a receptor cells, you get a depolarization in the cell. And you have to stop me because Mickey tells me that maybe I'm speaking about some things that are not clear. If something is not clear with what I tell you, please stop me and shake me up. Okay? So I assume you know what the depolarization is, what ions are, etc., etc. Anyway, one photon will cause a depolarization of a few millivolts. Let's say it corresponds to about 10,000 ions. And the question is, what is the price that we pay? Okay, so we put in one photon, 10 to the minus 18 joules, and we get back a few millivolts of depolarization in the receptor cell.
Which is more.
It's not more, it's much, much, much more. Okay, so it's not that one energy is transformed to another energy, but rather one energy triggers a cascade of events. Exactly, that's the point here, okay? So Eli would do the calculation, so you all know or should know from physiology how do membrane potential or the membrane potential of a cell evolves, and to do that we need the masters, which are the protein pumps sitting on the membrane, pumping sodium out, potassium in, for an exchange of 3 of 2 ions. And every cycle of this pump, which gives us 1 proton difference, okay, because the Na is plus and the K is plus. And to get 10 to the 4th or 10,000 ions through, you need this pump to work 10,000 times. And what Eli again found out is that the energy of a single hydrolysis reaction of these ATPases pumps is 10 to the minus 18, which means that you need 10,000 times more energy than you put in. That's the price that we pay.
It's not the action potential, it's not the sodium potassium pump.
The sodium potassium pump is the pump that is responsible for the gradient of the different ions across the membrane. Okay, so if you open the channels, there's going to be a flow of ions in, and then somebody has to maintain this gradient. The secret of maintaining this is these ATPases pumps, and we have to pay in the price of ATP. Okay, in biology that's the currency. Here we use shekels, in the US we use US dollars, in the body we use ATP. That's the price that we pay. We work hard for hydrolysis of ATP, and that's why we breathe and eat to get some energy so we can pay with ATP. Anyway, the basic idea is that the stimulus energy drives the receptor cells into electricity. So the language changes now at the receptor cells from the physical world, which can be electromagnetic waves or pressure waves or chemicals or whatever, and it changes into electricity, which is the language of neurons, and this is critical. Okay? This phenomena is called signal transduction or sensory transduction of the receptor cells. We have different types of potentials, different mechanisms for receptor potentials. We will see direct mechanical coupling, for example in somatosensation or hearing. where the actual physical pressure waves deflect the hair cells, and this physical deflection causes an opening of receptors on the membrane. This is true in hearing, for example, or in somatosensation in some aspects. We will see ligand-gated channels, like taste, that are opening receptors, and in many cases we will see second messenger cascades, like in smell, in the sense that I will teach, and in vision, okay, that the photoreceptor causes a cascade of transduction in the cells. These are all plotted, or generally plotted, in table 21.1 in chapter in the Candel book, and it's important that you get acquainted and know these different senses. Who does what type of mechanism is involved in transducing the physical energy into electricity. So like I said, this is an example of direct coupling, of mechanical coupling of the hair cells. Eli will talk about the mechanism of hair cell transduction more. He will elaborate on that. The second cascade example is has been solved to a large extent with the cell biology, and we know, from light until the different molecular cascades that induce the hyperpolarization in this case. If any of you was last week in Baruch Minke's talk, so you know that we now know the system quite well. Okay, so these are just examples of different transduction variables. Okay, the next thing is spike generation. Yes, okay, so some receptor cells which sit in the olfactory periphery will themselves, you will see changes in electricity, some receptor potentials, but they will not necessarily spike with the spikes, but they will, in some cases, they will synapse on other cells, which will then spike and transfer the information to the central nervous system. In some cases, they will spike. Like, for example, in smell, in olfaction, the actual receptors that, the receptor neurons that we have in the epithelium will actually send an axon into the CNS, and we can record spikes from these cells. Okay, so spike generation is not necessarily something that you will see in the periphery. Okay, one important thing that we will talk about now is that neurons is the first welcome to the physical variable in the world. And these neurons are filters, and they are both spatial filters and temporal filters. What do we mean by that? First of all, neurons that are sitting in the periphery will be sensitive only to a given part of the physical world around us. It's not that we have neurons, although they are responsive to electromagnetic waves, they will not respond to all the electromagnetic waves throughout the spectrum. Take vision, for example. If you record from different cells, let's say we have, as you may know or may not know, and if you don't, we will get acquainted, that the eyes, for example, have 4 different types of receptors, 3 cones, 3 different cone types, and 1 rod. And these have different sensitivities. For example, the cones, there are the red cones, the green cones, and the blue cones, which are different types of receptors in the retina. And this means that these cells respond, for example, the red cone responds to light only from, 475 and up to 660 or so. But you see that these responses, what is plotted here is the stimulus, the wavelength that the neuron was stimulated with, and the magnitude of the response of the cell. You see that each neuron has these so-called receptive fields or tuning curves. And this is also a very basic principle in sensory systems or in sensory neuroscience, is that single cells will have receptive fields, which means that they're responding only to a given part of the physical world around them. Together, they cover the whole spectrum. Okay, so for example, the blue cones will respond to electromagnetic waves in the lower values. the green higher and the red higher, and together they will code for the whole spectrum. We do not have any sensors that will code beyond 700 nanometers, therefore we cannot see in the IR. Okay? And similarly in the UV we cannot see. So this is actually a physical limitation of our system, that our sensors can sense only a small part of the spectrum. Of course, the problem arises is that a single neuron cannot code for the whole information. Okay, let's take the red cone, for example. If we plot how much electricity you get for a given wavelength, you see that because the nature of these receptor fields, you will get the same number of spikes from this cone, from this cell, for 620 and for 500. So of course we cannot differentiate what is the stimulus exactly based on a single cell or a single receptor, but rather we need the combinatorial code of both of them of different types of cells in order to extract the meaningful information from the receptors. Okay, this concept of receptor fields is not exclusive to the periphery, not to receptors. You can find receptor fields throughout the nervous system. This is an example of 9 neurons that we recorded in my lab from the auditory cortex. On the y-axis is frequency, on the x-axis is time from 0 to 200 milliseconds, and we give a very short beep at time 0. In this case, actually, it's from -20 to 200, and this shows the spikes. This is a normal way to represent responses. These are called raster plots, where you show exactly the space of or the region of the spike for different stimuli. You see that some neurons have narrow receptor fields, some neurons have wide receptor fields, and so on and so forth. You will see throughout the course that we like to present both raster plots as well as the post-stimulus time histogram, or PSTH, which is basically the average of the raster plots. This is specifically important when we look at neurons in higher brain areas, because in higher brain areas, neurons behave quite differently than receptor cells in the periphery. In the periphery, we have rather high fidelity of the stimulus. The neurons the sensor will sit quiet, and the stimulus come in, they will respond rather reliably. So if you go higher into the nervous system, this relates to what's your name?
Adi.
Adi. I should remember this name. I think I will from now on. So that Adi doctor to me is that there's more trial-to-trial variability if you look at neurons in the CNS. This is why we like to show raster plots, and we show the different repetitions. And this has consequences when we speak about how populations of neurons code information, and I don't know if we will get into correlation of noise and things like that. We also measure things like thresholds. I was too lazy to turn this into a white slide. So basically we record the raster and PSTH of a neuron, and then we can plot how this neuron responded to the different frequencies. We plot these in some color code. And then what we do is we decrease the level of the intensity and do the same thing again, and do the same thing again, and do the same thing again until this is decreasing the intensity of the stimulus until we get these plots of threshold where we can determine the threshold responses and the best frequency of this neuron, for example, okay, which will be around 14 kilohertz and 24 dB SPA. Okay. Sorry. Okay, so neurons have receptor fields, and we can perceive the stimulus by varying combinations of the receptor potentials. This is again something I just plotted in to show you that what a mess it is up there. These are neurons responding to natural stimuli in the auditory cortex. So you give a song of a pup to its mother and record the spikes, and you see that some neurons respond only to this call number very reliably to call number, I don't know, 8, some only to call number 5, some respond to all all the syllables in the call, etc., etc. So you will see when you go higher into the brain, things like context and history of responses in a very nice way. Okay, this is will become evident, for example, in Ellie's part, which is something that Ellie is actually studying in his own lab. Okay, what types of tools do we have to measure physiology? This whole issue started from people like Vernon Mountcastle and Jubel and Wiesel, which you are hopefully started to read their classical paper from '62. Actually, in their introduction, they already give compliments to Mountcastle on his studies on the somatosensory system. And this is classical stuff from the 1950s, things also like Bert Sakmann and Erwin Neher, which developed the patch clamp technique, which allows us to look at single channel activity. And this is the beginning, and we're now, again, you guys are in the middle of a revolution in terms of technique. It's actually really unbelievable to compare how, when I was at your chair doing graduate school, the tools that I had available to the tools I have available now in my lab. What you see here is a mouse running on a ball in a virtual reality, okay, while they image from its brain. And this is very different than what we envisioned that we can do even 10 years ago. And this is continuously changing, okay? Classical electrophysiology, you will see expressions like single channel recordings. This is the patch clamp technique that Sakmann and Neher got the Nobel Prize for. Spiking activities, this can be recorded intracellularly, which is the gold standard, or things like Mickey London is doing in his lab. And we can record potentials, electrical potentials from the brain extracellularly, and this goes more coarse and coarse, you know, things like local field potentials, EEGs, etc. The advantage of using electrophysiology is that it's the real language between neurons. They actually, and it's exactly this place of the receptors which hits reality that has transformed the language to electricity. And electrophysiology is the gold standard. Recent, more modern techniques use things like optical physiology. I showed you that image from Tank's lab on the mouse running on a ball, and you can put them under a 2-photon microscope to look at optical changes of calcium ions, which have some correlation to the physical activity. I thought I'll have time. Okay, I will. 1 second, just to show you how this thing looks like in reality. So this is again data from our lab where we inject the brain with a fluorescent dyes that report physiological activity. Right? And now we zoom into the brain while the mouse is alive, and you can see astrocytes in red and neurons in green. And then we can record in the next slide, many cells. So this is an example, I think, from 16 cells simultaneously, where you can actually now record population of cells and see not just how a single neuron responds to a stimulus, but rather how a pool of neurons or groups of neurons encode sensory stimuli as a group. Which is, again, this is all rather new technology. We're still in early days of this, but these are the new emerging technologies which you will see in labs like Mickey's, like mine, and other, where we are bringing in new technologies of this sort. Okay. This is now, I wanted to give you a little bit more, all kinds of miniature 2 photons that are implanted on rats. People can now, image while the animal behaves, actually, etc. But eventually, at the end of the day, what we want in sensory physiology is, again, you should keep on in mind that what we want is to link sensory perception to the physiology. And in this case, I showed you this, I think, earlier on, where people have to estimate how deep was the indentation on their skin. And if you record from the amplitude of the indentation and record, sorry, I scanned this, but it came out, it's just the number of spikes And you can see that this receptor cell is in fact coding the amplitude of the skin indentation. Okay? That's very easy to see that the physiological parameter and the physiological response are linearly matched, and this matches nicely the perception. And these are the type of experiments that make us think that we know how or what is the role of the cells in the periphery. This is far from the truth when we go deeper into the sensory system. But let's stay in the periphery. In the periphery, you will find receptor cells that behave not only as the one I showed you in the slide before, where the magnitude of the intensity increases and you get more spikes, but you get all kinds of wonderful combinations and different coding schemes of the spikes from the cells. One example that is nicely given in the book is these rapidly adapting receptors. Basically, these are these cells are reporting when is the stimulus turning on and when does it turn off. So if you give it a step pulse, the neuron will spike in the beginning and will spike in the end. If you give it a ramp, okay, so it takes some time until the stimulus comes on, then while the stimulus comes on or is changing, the neuron will continuously fire. If you lower the ramp, the neuron will keep on firing until it stops. So basically it reports change, and this is a conceptually different neuron than this type of neuron, which reports, by the number of spikes. You will get familiar with concepts like rate codes and things like that. Okay. So let's see, the fidelity of the stimulus. Okay. How well do our receptors actually mimic the world around us? And there are 4 things that affect the fidelity of the physical stimulus that our receptors report back. One is physical limitations, actual physical limitations. The second is spatial filtering. We will dwell a little bit on that. Just briefly mention temporal filtering and noise. Okay. First, physical limitations. These are obvious. We can't see, things less than 1 photon. We cannot hear things which are lower than the thermal noise. And there's so much limitation on our chemical senses. What?
What is thermal noise?
Thermal noise is that there is movement. Basically, when we will study about sound, you'll see it's pressure waves in the air, and the noise, the basic noise, if you can measure movement of the air without any stimulus that comes on. Okay, so this is thermal noise, and this is basically the noise level in the air. And you cannot hear things which are lower than the thermal noise. It's a matter of signal to noise ratio. Molecules, there is a range of things. We know much less about how much, for example, in olfaction. The highest world record is some Japanese studies from the, Department of Agriculture in Japan, that they found that humans can detect parts per trillion of a chemical, which basically means you take a swimming pool, an Olympic-sized swimming pool, put 1 drop of chemical, and the person can distinguish between this pool with the drop and that pool without the drop. But this is quite unique. Most of the sensitivities in olfaction are parts per billion, and mouse can do that very easily. Humans can also do parts per billion. But basically, there's a physical limitation that we cannot. And the physical limitation is things like we can't see, things from behind and things like that, because just we're limited physically by the physical machinery that we have in our biology. The second thing, which I will dwell upon in the next 10 minutes until the class is over, is spatial filtering. Then this is divided into different parts. First of all is receptor fields. Of course, a neuron will sense only the region that it occupies. For example, it will have receptors on a given area of the skin in somatosensation. It will be in a particular region on the retina for vision, and it will be able to hear specific frequencies in the hearing domain. Generally, we refer Normally, when we say receptive fields, we refer to the relevant dimension encoded topographically on the sensory epithelium. Okay? So on somatosensation, we say somatotopy, depends where do the receptor resides on the skin, and there's the topographical organization in vision and tonotopical organization in hearing. If you go to chemical senses, this concept breaks down because, for example, in the epithelium, our receptors are mixed and matched and they're all over the place, and there's no real meaning for location. But certainly in somatosensation, vision, and hearing, the location of the receptor on the epithelium plays a major role in our understanding how the information is coded. Okay, so the most intuitive example are the receptors on the skin, which filter the information spatially because simply they reside in a given region on the skin. The spatial distribution of not only the active but also the silent receptors provides a sort of a neural image of the size of the stimulus. If I give a stimulus in these two spots, so this neuron and this neuron will be active, but in order to know that nothing is happening in between, these have to be silent. So the silent cells are also a part of the neural image that now the brain sees when it talks to the receptors. Okay, that's one thing. The other thing is that the neural representation of a scene is composed of many receptors encoding different aspects of the stimulus. Not everybody will encode, what or where. Some will recode where. Some will be more sensitive to when, like the receptors we saw before. Okay, the ones that are tonically active versus the ones which are phasically active. So all of these we'll have to take into consideration. Okay. So what is the resolution? We're still talking about the fidelity of the physical stimuli. How reliable do receptors capture the real world? And this, again, they don't rely. We saw this very early on. We don't have a perfect reflection on our sensors of the environment around us. And this brings me to the issue of resolution of the system. The first thing that is important in the resolution that we can capture is the number of receptors on the sensors that we have, as well as the organization of the receptive fields. First of all, let's see the number of receptors. This is very intuitive and clearly defined when you look at things like pixel resolution in an image. In this case, we have 400 receptors. Each pixel has exactly the same dynamic range. But the image is blurry, and the more receptors you plug into the system, the crisper the image is. And we also have these types of different densities of receptors in biology. For example, the receptors in somatosensation, we will see that there is a higher density of receptors on the fingertips. This is why blind people read with their fingertips and not with their elbows or arms or the back of their hands, etc. This is some cartoon showing how sensitive or how our organs look like based on the number of receptors or density of receptors that we have. Again, in the palms of the hands, especially the fingertips, the lips, and the face are very sensitive in somatosensation. This is also true for the retina. We have a region, the density of receptors is different in different regions of the retina. We have a special unique spot in the retina which is called the fovea. And the fovea has the highest density of receptors per millimeter square per region. And indeed, the relative acuity of the fovea at that region is the highest. So if we read, okay, and we want to take details of the letters and we do precise things, we are using our fovea to do that, and less so the other parts of the retina where we need less resolution when we look afar, for example, or on scenes or something like that. Okay, but again, we will talk about the visual system more closely. I brought back this image because one thing which is clear across sensory systems is that the first step of the receptors in the periphery is to break down or deconstruct the stimulus into its basic components. You will not see neurons responding to Bart Simpson in the cochlea. Okay? You will show the word Bart Simpson like I showed you last time. You can't see things like that. The first thing that the sensory system is breaks down, takes a big hammer and breaks down the physical stimulus into its basic component, and then we start to build things up. So in the visual system, which is perhaps the most well-studied system in sensory physiology, especially in higher brain regions, if you put your electrode in station number 4, you will see neurons responding to lines or moving gratings. If you put it higher on, you will start to see more abstract structures, etc., etc. Okay, so there is a bottom-up hierarchy, although this, like I showed you before, can contradict with our intuition about perception. We can, in fact, in pop-out tests, identify the pop-outs in higher brain regions faster and easier than we do the more fine details. Okay, first question comes up is how much receptor fields, how much should receptor fields overlap? Clearly, you can imagine that we have a high density of receptors, but their receptor field is so wide that they are not very informative. Of course, the best resolution that we'll get if we'll have many receptors with small receptor fields, and still those have to overlap to some extent. And the answer to this comes from a basic engineering or data acquisition modes. I think Eli is teaching you data acquisition, right? Or what's the name of this course? Ibud Meita? Information processing, yes. Okay, so in order to, this is something called the Nyquist criteria, that in order to capture the frequency, the maximum frequency of a signal, you have to sample it twice as high, minimum, to capture the frequency. If you want to capture both the frequency and the amplitude, you have to go much higher than that, than twice the frequency. But in general, if we are talking about resolution and the overlap, the receptor fields in biology or in the sensory systems overlap by half, roughly, and that's what they should, in order to capture the maximum frequency of the receptors. Okay, so in order to capture this distance, let's say this is 10 microns, in order to capture 10 microns, they have to, the receptor fields have to be positioned like this. So there must be a twice overlap than the frequency that you want to make. This is exemplified. This is classical basic engineering. There's for example, here it's a 5 megahertz sine wave, and we sample it at 1 megahertz. Okay, so these are the points of sampling, and if we would have to reconstruct the signal, this is the sample that we get. Okay, this is called aliasing, and aliasing the signal is bad. It normally gives us a lower frequency as compared to the real frequency. And in order to capture the correct frequency, let's say 5 megahertz, you want to sample at 10 megahertz. Okay? So if the rate of change of the stimulus is bounded, most of the signals that we hear are band limited, so at some frequencies they will fall to 0. Then we need to sample at least twice the highest rate of the change to get a perfect reconstruction of the frequency. Okay? But again, not the amplitude. In order to get both frequency and amplitude, we have to sample higher. And you can go to the books, Nyquist request. This is again very basic. I wanted to show this in the context of the resolution of the visual system. So in order to resolve 2 points in space, the definition of this is that we will be able to distinguish perceptually that there are two dots here and not one. Of course, if they will go closer and closer together, you will lose the resolution, and the resolution is defined on what is the distance or the shortest distance that we can define two objects in space. Basically, when a spot of light falls on some surface, it will produce point spread function and will not be a perfect spot of light, but rather will have this unique profile. And basically the resolution is defined as that the peak of the second spot will have to fall on the trough, on the first trough of the first spot in order to be able to resolve these two issues. This is classical in optics. I don't know if you're getting anything in optics, but basically any spotlight that goes through a pinhole will have these what we call airy disks. So we'll have a bright spot in the middle, but then because of diffraction of light, we'll have dark bands around it. This is an image of an airy disk, a classical airy disk. And of course this sets physical limits to the resolution that we can identify between two different points. Again, Eli went the whole distance to do the calculation with you. Let's say there is a point at infinity which shines coherent light onto the eye, and assuming that the opening of the pupil of the eye is 6 millimeters and the size of our eye is 1 centimeter, the question is what is the spot at the retina? Okay, and one can calculate it based on the details that are shown here, which we don't have time to go through. But basically, if you know the wavelength, notice that this is 600 in the calculation, but this should be yellow line and not blue. But just yellow doesn't show nicely on the board. Basically, you can calculate that a spot in infinity will end up at a 1 micrometer diameter on the retina itself. And in fact, when you go and look at the receptors at the retina, you see that at the fovea, the highest density of receptors, we have about 200,000 receptors per millimeter square, which is roughly a photoreceptor every 2 micrometers. Okay? And 2 micrometer, every 2 micrometers, and if those will overlap by half, this means that we get about 2 micrometer resolution at the retina. Okay? And you know, this is again from last year's talk to show that we are a little bit lower than that. The book says 200,000 receptors per millimeter squares. If you look at the scale bar here, if it's correct, we get somewhat lower than that. And of course, our resolution is lower. There are other distortions in the eye. The resolution, in fact, is much lower than the perfect resolution. But this is the perfect resolution that we would be able to achieve if we consider only physical constraints on the retina. Okay? So if we're looking at the physical constraints, first of all, the sampling that we have, even in the fovea, is lower than the rate that we should have if everything would be perfect. But again, there are other distortions in the eye, so we are in the ballpark, roughly. And okay, it also is, of course, it matters how good the eye is. For example, the size of the eye. Size of the eyes of humans is very different than the size of the eyes of birds, for example. In fact, when we check what's the resolution of birds, they are as good as humans. Humans have the best, almost the best known visual system in the animal kingdom. Birds are as good as them, which is quite remarkable because their eye is much smaller and their physical constraints on the resolution is much more challenging, and they do this by having a substantially higher density of photoreceptors. But in real life, at the end of the day, the performance is as good as humans. Other examples are how well we can differentiate two tones in the different ears. We have a big head, so this should be an easy task. Some animals have much lower head size, and it's again quite remarkable how their systems is in a way better per size of the eye or size of the head in the performance. Last thing which you will, so this is the absolute resolution, how well we can differentiate between two spots or two sounds, etc., etc. Some senses have what is called hyperacuity, which is different than acuity. Acuity is the resolution I talked about, how we can differentiate two different stimuli. Hyperacuity is in fact the ability of the sensory system to differentiate signals which are have much higher resolution than the sensors themselves. So for example, if you look at receptor cells in the cochlea, their bandwidth is about 1/6 of an octave, which is about 130 hertz at 1000, at 1 kilohertz. But the frequency resolution that we can detect is 5 hertz, okay? And the same thing is with ITDs, with inter-oral time differences, but you will discuss these later with Ellie. Color vision also works with higher acuity, so we can get super resolution, so to speak. We can do better than the resolution of our sensors. Although somatosensation, for example, and also spatial vision are limited by the Nyquist rate criteria that we talked earlier. So you will hear about things like hyperacuity, which is some things which are rather incredible how we can differentiate. In ITD, for example, is a beautiful example of hyperacuity. Last and not least is temporal filtering. This is fast, huh? Is the temporal filtering. Obviously, the neurons are limited. Okay, they're limited from above and limited from below. Above, we're limited by membrane time constants of the cells, transduction processes, okay, how long it takes protein to be shuttled from on the membrane and to activate another protein. And we're also limited, what Ellie calls from below, by spike rate adaptation, synaptic processes of synaptic depressions from the biology itself. Okay, so these obviously induce limitations to what the neurons can or cannot detect. Of course, we cannot do much better than our sensors, besides some tricks in hyperacuity. Okay, these are different types of receptors. These are called tonic, when they fire tonically. We saw that before. Phasic is when the stimulus is still on, but the neuron adapts and stops firing. She is right. I am finishing up. You see, she's right on time. She should draw when I have to go. Okay, this we can learn. Noise, of course, signal to noise. We can do better than our noise. Last slide of the talk is that a canonical, if you look at a circuit in the brain, they are not simple machines to relay information onwards. Okay? Neurons in any circuit will have converging information, diverging information. If you look at almost any neural circuit in the brain, there will be afferent inputs into the system. These are expressions that you should know at this stage. Projection neurons are neurons that take the information out of the circuits. Local interneurons are neurons which are seated locally throughout their cell body, their dendrites and their axons, are local in the circuits. Many of them will be part of all kinds of inhibitory loops. And if you normally look at a receptor in the periphery, which we understand quite well, it will basically have an on-off switch-like behavior. Okay, so it'll be quiet and will turn on tonically or phasically, whatever, but nothing too complicated. Another thing which, because this is why we also understand these cells better. If you look higher in the nervous system, inhibition kicks in, and things are much more complex than neuromodulation, etc. Although you can find some of it also in lower stations. With this, I'm going to end my talk on sensory physiology exactly on time, -2 minutes. And next Wednesday, we're going to talk about 2 papers which you will read. One which I will present, the second which we will discuss. And then we dive into the pool with Idan Segev on single neuron computation. Have a nice day. לילה מהיר.
זה לא הזמנים
עוד אין לי, אני אעשות, אני רק רציתי לראות כמה אנשים יש לי בכיתה, מי רשומים
של השעות של...
standard of calibration worldwide that is used to calibrate a given level of sound, intensity of sound. When I say 72 dB decibel SPL, sound pressure level, it is related to some kind of scaling. There's other types of scaling that other people use, okay, not SPL. But the standard in our field is SPL.
So does it have to do with what pressure, atmospheric pressure that you decide?
It's some kind of calibration that I don't know exactly how they do. You know how they exactly do the SPL calibration? I don't know.
It has something to do with pressure. It's a measure of pressure. And then you kind of make everything in dB, meaning like log scale compared to the pressure. So basically, if I give you like 1 kilohertz, when they say that the sound level, there's a sound level at traffic code shouldn't be more than such and such. And you have to compare it to some units. So there are like these devices that you can buy, and they will give you exactly this level that you have to record at the, you go to the road, you record with your microphone, but then you don't know how to calibrate, and then you go to the lab, you record 1,000 kilohertz with this device, and then you can compare it relatively exactly.
It's a reference measure of the intensity of sound.
This is actually a physical measure of pressure.
But there are a few different ones. So if you look at, I think audiologists also use DBSPL in the clinic, but there are others who use different measures of sound intensity. But you will see in the neuroscience community, this is the standard measure that we use for sound intensity. DBSPL. Exactly the nature of it, I don't know. I have to ask you.
It's also very tricky, no?
What?
Very tricky, because when you put like two mics, how do you know what they get? You can say what you play, but not say what they get to the ear. Right?
Yeah, you need to put microphones where their ears were supposed to be or something like that. But, we think
You calibrate it well.
yeah, it's like a standard thing. When you buy a machine, you get a calibration kit with it. So it will, it's just like a reference measure of intensity.
I got that point. It's a sense of reference. But I was just wondering how would you calibrate it with pressure really?
I don't know. I don't know. I have to look at it. I don't know exactly.
I think I read it on even in Wikipedia. Pretty good way to. They would just.
Okay, I will look it over.
I'll show you in a second.
Two more questions.
Okay.
Well, you have the slide, and
Yeah,
if it
maybe
comes up, you can either stop by or drop me an email.
And another thing is, so I happened to come to the last semester quite late because I got my letters late and all that. I came in December, so I'm supposed to be giving my exams from the last semester now.
Oh, okay.
The first time in.
So you want to be towards the later in the semester?
Yeah.
Yeah, sure.
Yeah, and also, the paper coming now on Wednesday.
Yes.
Yeah, I might not be able to.
Okay.
So because Monday I've got an exam.
No problem. This is demo. Okay.
Yes. 20 microPL RMS, which is usually considered a special for human hearing in microphone kilohertz. So this is like PA. Let's give you Australis.
Yeah, I think it's if I looked at it many years ago. So it's some kind of, I think, perceptual measure or something like that they calibrated to. Whatever. Anyway, it's a reference point to intensity, and we can't hear below, I think 50 or 40. We can't really hear very well. Okay.